We'll enter into the phylum Mollusca by looking at some gastropods like this wavy turban snail, Megastrea undosa. We'll organize them according to this slightly simplified gastropod phylogeny, in which there are four major groups. One clade composed of patelogastropods and Veda Gastropods, which we'll look at today, and another group composed of cenogastropods and heterobranchs, which we'll look at next time. All members of the patelogastropods and Veda Gastropods that have ctenidia have bipectinate ctenidia, and almost all of them mate by free-spawning gametes into the water. We'll focus on the anatomy of one species of patellogastropod, Laudia limacula. In ventral view, you can clearly see the distinction between the head and the foot. The tissue lining the shell is the mantle, and at its edges you can see many tiny palleal tentacles. On the animal's right, your left, you can see the tip of its single ctenidium. That ctenidium is attached on the animal's left side, so it's the left ctenidium. The tips of some of the teeth of the radula are golden in color. Those ones are mineralized with a combination of opal and an iron-containing mineral called goethite. Here you can clearly see the muscles on the animal's right side that connect the body to the shell. The ctenidium is sticking out of the region behind the head, which is the mantle cavity. You can see the same shell muscles on the animal's left side. Poking the edge of the mantle shows that it's not connected to the shell at its edge. It can be pulled back a little. When gastropods have only a single ctenidium, like Laudia, it's the left ctenidium, and they use it to generate an asymmetric flow of water through the mantle cavity from left to right. To see the mantle cavity more directly, I anesthetized an individual and removed it from its shell by cutting those shell muscles where they attach to the shell. The mantle tissue covering the mantle cavity got a little torn during that process, so we can already see into most of the mantle cavity. The whole dorsal side of the body is covered by mantle. In the center of the body, that mantle tissue is bright green. The mantle at the edge of the shell, which bears those pallial tentacles, is a little thicker. You can see it surrounding the left, posterior, and part of the right side of this animal. The head is red because the radula and odontophore muscles have myoglobin in them. I tore away the last bit of mantle over the mantle cavity, and now the whole mantle cavity is exposed. The ctenidium is very large, and when you turn it a bit, you can see that it is bipectinate. There are leaves of tissue sticking out on both sides of the central axis. If you pull back the tenidium, on the floor of the mantle cavity you can see two patches of red or orange tissue. These are the two osphadia, chemosensory organs. On the right side of the mantle cavity, you can also see the rectum. In this individual, there's a thread of feces emerging from the anus, so it's easy to spot. Harder to see, right under the rectum is a greenish bump that is the Nephritia pore. Here it is right between the right osphradium and the rectum. The pericardium and the heart are right under this little triangular patch of mantle at the base of the tenidium. If you tear that mantle, you expose the pericardium, the space surrounding the heart, and the heart itself, which is still contracting in this anesthetized specimen. I can't tell the difference between the auricles and the ventricle in this heart, since it's all the same tan color. Under this bright green mantle tissue is the visceral mass, digestive system mostly. Some components of the digestive system are poking out to the right as well. To see the anterior part of the digestive system, I cut into the dorsal part of the head. That exposes the bright red buccal mass, which is the Odontophore and associated muscles, and a flimsy orange-colored tube on top of that. That tube is the esophagus. If you remove it, you can see the radula in the radular sac below it. The radula is only attached by muscles at its anterior end. If you pull on the posterior end, the whole thing slips out of the body. Here you can kind of see that the Odontophore is made of cartilage, but that's a lot clearer if you remove some of the red muscle covering it. The radula slides over those two lobes of cartilage as it's being used. The anterior teeth of the radula are supported on a wide chitinous ribbon, so there's more surface area for radular muscles to attach to. Here's a dorsal view of the whole radula. You can see that it's made up of many nearly identical rows of teeth. I didn't count them, but expect that there are at least 100 rows in this species. You can also see that some of the teeth are dark and mineralized at the anterior end, but they are pale and unmineralized at the posterior end where they are being formed. One interesting thing you can see in a side view of the radula is that the anterior-most teeth, those in the first two rows, are shorter than those of the more posterior rows. The teeth in those first two rows are the teeth that the snail has been using, and their tips have been worn down with use. That's what happens when you scrape your food directly off of rock. In each row, you can clearly see 4 teeth with dark mineralized tips. Those are called lateral teeth. But there are more teeth than that in each row. You can see a pair of tiny, hook-shaped, unmineralized marginal teeth in each row as well, for example. There might also be more unmineralized teeth in this radula. We really need a scanning electron microscope to know for sure. But Laudia definitely has no more than 13 teeth per row. I think actually more like 7 or 9. Here's the anterior end of the radula, and moving backward to emphasize the difference in mineralization among regions of the radula. The newest teeth at the posterior end are not at all mineralized. To get one more view of the overall anatomy of Laudia, I anesthetized and preserved one to stiffen the tissue. I popped it out of its shell and bisected it with a razor blade. Here it is in section. My cut went right through the middle of the radula, so you can see its relationship to the mouth here. This is the ctenidium in the mantle cavity. You can also see the thin mantle tissue covering the mantle cavity. This is a soon-to-be fecal pellet in the digestive system. This spongy brown material is likely mostly digestive gland. This green tissue with little spheres in it is a large ovary containing eggs. Right below it is the muscular foot. And one more thing, the shell. You're all familiar with intact limpet shells, but we can also look at it in cross-section. To do that, we need to fracture it. Here I've mounted it on clay so we're looking at a fractured edge. We can see at least 3 layers: an outer brown layer, the periostracum, a thick layer of crystals oriented in various ways, and a thin inner nacreous layer on the bottom. The periostracum also has a lot of other material on it: sediment, algae, that kind of thing. And now Veda Gastropods. These are similar to patella gastropods in lots of ways, including having bipectinate ctenidia and being mostly free-spawning. But they have more diversity in shell form and in number of ctenidia, as we'll see. Some have 2 ctenidia, one on the left, one on the right, while others, like Integula isoni, have only a left ctenidium. Some Veda gastropods have an operculum on the back of the foot to close off the shell aperture when they pull into the shell. Integula's is round and thin, made of chitin. Integula has 2 cephalic tentacles and many tentacles on the edge of the foot. Those are called epipodial tentacles. The cephalic tentacles are a little longer and thicker than the epipodial tentacles. The shells of these 2 individuals are covered with serpulid annelids. You can see the 2 cephalic tentacles on the right in this individual. They behave differently from the epipodial tentacles. This individual is walking away from the camera, which lets us see how it balances the shell over the foot. In ventral view, you can see how this snail uses its foot to walk. To see the mantle cavity, I anesthetized a tegula and removed it from its shell. All the spiral-shaped bits on the left and bottom are visceral mass, including the green gonad. The mantle tissue is intact in this animal, hiding the mantle cavity. But if you peel it back, out pops the single tenidium, the left tenidium, and the rectum, which looks like it's full of a fecal pellet. I did not identify an Osphradium Integula. We can find the radula like we did in Laudia by cutting into the dorsal side of the head. Here it is, much shorter than that of Laudia and with no obvious mineralization at all. There are also way more teeth in each row than in Laudia. Laudia had no more than 13 per row. Tegula has at least 40 or 50 teeth per row. Many of those are the thin, brush-like ones on the left and right margins. This radula is apparently mostly used for sweeping up diatoms and other loose, tiny particles into the mouth, as opposed to gouging out hard material like in Laudia. Next, the Veda Gastropod Megastrea undosa. This animal is very similar to Tegula in terms of its mantle cavity, tenidium, and radula. One difference that you can see externally is that the operculum is white. It's made out of calcium carbonate. Like the radula of Tegula, Megastraea's moves in a complex way. The ribbon rolls up into a tube as it passes over the Odontophore back into the radular sac. You can also see a pair of white upper jaws in the mouth of this animal. These are not part of the radula at all. And now for a Veda gastropod with two ctenidia, the giant keyhole limpet Megathura crenulata. This animal can extend its mantle out of the shell and reflex it back onto the shell so that most of the shell is covered with tissue. The sole of the foot is this nice yellow-orange color. It also has a hole at the apex of the shell. More mantle tissue can protrude from that. The mantle tissue covering the shell is defensive. It makes it hard for predators like sea stars to grab onto, and it may also secrete some unpleasant chemicals. Here's an empty Megathura shell. If you look down into the keyhole, you can see two white structures. Those are the two ctenidia. Megathura has one left ctenidium and one right ctenidium. Megathura is not very fast, so I sped this up 3 times so you could see locomotory waves on the foot. They are very different from those of Tegula. The hole in the shell is used to channel outgoing mantle cavity flow. The anus is positioned right next to the keyhole, so feces can be shed directly to the outside. More generally, water that enters the mantle cavity and passes over the ctenidia then exits via the hole. This is really different than Laudia, Tegula, or Megastraea, where flow goes from left to right through the mantle cavity. It's what you can do when you have some plumbing built into your shell. I collected our final Veda gastropod at one of my favorite stores. I don't know any other grocery store that stocks live abalone, and at an excellent price. I bought one to bring back to our marine lab as a pet. We already have 3 others that originally came from H Mart. Though they're labeled black abalone, they are red abalone, undoubtedly from one of California's abalone farms. Abalone love to eat kelp, and H Mart sells kelp too, so it was really the perfect store. Red abalone often sit in wait for drift kelp, and when it comes by, they grab it with 2 muscular lobes of the foot. Acquiring a piece of kelp is obviously very exciting. Like Tegula, abalone have lots of epipodial tentacles. Here's another one trying to feed, showing off its foot lobes. When not feeding, they often wait in this position, just hoping for a piece of seaweed to drift by, which they can then sense with their epipodial tentacles or with those foot lobes. And one more. Abalone have two ctenidia like keyhole limpets, and they also have holes in their shells, but more than one. Like keyhole limpets, water that enters the mantle cavity passes over the tenidia and then out of some of those shell holes. Here's an abalone out of its shell to show you that mantle cavity and other parts of its anatomy. This circle is the muscle that connected the abalone to the shell. This is the visceral mass, which includes most of the digestive system and gonads. And this is where the heart is located in the pericardium. The mantle has a slit in it underlying the row of holes in the shell. Inside you can see one bipectinate gill on the left, and one on the right side. And at the back of the slit, you can see the rectum. Nephritia pores are also present there, but we can't see them in this view. So when water passes through the mantle cavity, it exits via the shell holes, carrying feces and urine, and when the time is right, gametes. You can see that the open holes in the shell overlie the mantle slit, so they're available for water flow. Here's an isolated shell. You can see about 5 open holes in this shell, plus one new hole that's in the middle of being made. As the shell grows, it adds new holes at the growing edge and seals over old holes at the back end of the mantle cavity. You can see where the snail has filled in old holes either from the inside of the shell or the outside. The spiral of filled holes allows you to understand how this shell grew, spiraling around from this spot, which is about where the original juvenile abalone shell was located after metamorphosis from the larval stage.